This video was made for a genetics class at Ohio Valley University and will cover the topic of the discovery of DNA as a genetic material. Before 1928, there was not much known about the structure of DNA. In 1869, Friedrich Miescher was able to isolate DNA and discover that it was an acidic substance which was found in the nucleus of cells. Miescher also discovered in 1914 that DNA could be stained, and as a result, he concluded that DNA of all eukaryotic cells were contained in the nucleus. In 1928, Frederick Griffith studied two different strains of Streptococcus pneumonia. The S strain is smooth and secretes a polysaccharide capsule, which allows the bacteria to escape attacks by an immune system. The R strain is rough and does not secrete a polysaccharide capsule. Griffith tested two different strains on mice. In trial A, Griffith used a live S strain. After injecting this strain into the mouse died within a couple of days. For trial B, he used the live R strain. After injecting the live R strain into the mouse, it showed no signs of any problems and survived. In trial C, Griffith used a heat-killed S strain. After injecting the mouse, the heat-killed S strain bacteria, the mouse was able to survive. Trial D was the heat-killed S strain and a live R strain. After injecting the mouse with both of these, the mouse died within a couple of days. Extracts from the tissue of the mouth showed live S bacteria. From this experiment, Griffith was able to come up with a few conclusions. The type R strain bacteria must have acquired the ability to make a capsule from the dead S strain bacteria because the live type R bacteria did not kill the mouse. Griffith used the term transformation for what had happened between the S strain and the R strain bacteria. He knew that a genetic material was the cause, but was unsure of the exact material that caused the type S bacteria to transform the type R bacteria. Oswald Avery was a chemist who studied pneumococcus, which is a bacterium that causes pneumonia. In 1936, Avery noted that the DNA did not seem to be a protein or a carbohydrate, but instead was a nucleic acid. His death in 1955 came before widespread recognition of his role in discovering the significance of DNA. Avery, McLeod, and McCarty realized that Griffith's observations could be used as part of an experimental strategy to identify the genetic material. At this time of the experiment in 1944, researchers already knew that DNA, RNA, proteins, and carbohydrates were components of living cells. To separate components and determine if there were any genetic material in them, they used biochemical purification procedures and prepared bacterial extracts from type S strains. They discovered that only one contained purified DNA, which was able to convert the type R bacteria into type S. When extract was mixed with type R bacteria, some of the bacteria were converted into type S. If no DNA extract was added, no type S bacterial colonies were observed. To verify that DNA in the extract was responsible for the transformation, Avery, McLeod, McCarty treated samples of the DNA extract with enzymes that digest RNA, DNA, and proteins. These enzymes were DNase, RNase, lipase, carboase, and protease. When the DNA extracts were treated with RNase, lipase, carboase, and protease, they still converted type R bacteria into type S. This indicates that the remaining RNA or protein in the extract was not acting as a genetic material. When the extract was treated with DNase, it lost its ability to convert type R into type S bacteria. This means that the deprivation of the DNA in the extract by DNase prevented conversion of type R to type S. This is consistent with the hypothesis that DNA is the genetic material. They successfully identified DNA as the transforming principle responsible for specific characteristics of bacteria. In 1947, Oswald T. Avery won the Albert Lasker Award for Basic Medical Research. Then, in 1994, McLeod McCarty won the Albert Lasker Special Achievement Award. Alfred Hershey and Martha Chase knew that bacterial viruses were very simple and made of only proteins, which were the outer shell, and DNA, which was in the inner core. They came up with an experiment known as the Blender experiment. In this experiment, they had T2 bacteriophage and E. coli bacterium cells. They used radioactive sulfur to visibly see the proteins and radioactive phosphorus to visibly see the DNA.
The two types of radioactive bacteriophages were added to separate cultures of bacteria. The phages infected the bacterial cells and seized control of the reproductive mechanisms in the bacteria. Hershey and Chase used a blender to separate the different parts of the mixture, lighter phages on top and heavier bacteria cells on the bottom. DNA was found among the bacterial cells on the bottom solid portion, which is called the pellet. Proteins were found in the upper liquid portion, named the supernatant, with the phages, which are dislodged virus particles. They were able to conclude that DNA was the material used in the transmission of phages and the creation of bacterial viruses. This is also proven by the observations of the phosphorus phages offspring because the radioactive DNA was found while the radioactive proteins were not. Prior to this discovery, the overall belief was that proteins were the genetic material. In 1952, they published their discoveries in a short paper. This paper then became the roots of the genetic research on DNA. It concluded that DNA was the location for life's hereditary data, and also that genes were made of nucleic acid DNA. In 1969, Hershey won the Nobel Prize in Physiology or Medicine for his findings on the properties of DNA. Because Chase was only Hershey's lab assistant during the experiment, she was left out in recognition for this experiment.